All right, well, it's 10 o'clock in beautiful spring Montana, and we'll get rolling. I'm Christoph Lambert, the Chairman and CEO of Golden Helix, and I'm so glad to welcome you today to hear all about learning from our GWAS mistakes from experimental design to scientific method. It's, of course, the topic of a recent paper that I published in Biostatistics with co-author Laura Black, and also the topic of a blog that introduces the paper, and perhaps some of you have read it. We had over 300 people sign up to this webinar, so it appears it's a very interesting topic to many. And so without further ado, I'd like to get a few technical things out of the way and then get rolling into the talk. So first of all, there is a Q&A that we'll have at the end of the presentation, and the way to ask your questions is by clicking the question and answer pane on the GoToWebinar view. So our agenda today is talking about the presentation, the paper, learning from our GWAS mistakes, and why should we care about that? Talk about the impact of poor study design. I'd like to talk a bit about a case study with a Sebastiani retraction of the exceptional longevity paper and what we can learn from that, and make some study design recommendations. And then, in keeping with the broad sweeping theme of the paper of looking from everything from experimental design to the scientific method, I'd like to revisit the scientific method because it turns out lack of knowledge about the scientific method may well be one of the sources of the problems we see in terms of the mistakes being made in GWAS and other areas of biostatistics. And then we'll close and have an opportunity for questions and answers. So here's the paper I co-authored with Laura Black, and she's a professor at the local business school university. Why business school? Turns out Laura Black is a world expert on system dynamics, and she spent a lot of her career looking at the systems in which we operate. And so an ongoing theme that I've been talking about in my blogs, and if you ever meet me face to face, I'll probably talk to you about how can we do our research better? What is it about our process that needs to be improved so that we can make more discoveries and advance human knowledge and improve human health? And so if you think about the process that we operate in academia and to some extent in government and in industry, we care very much about significant findings. Probably what drew us to science is we're passionate about doing research, doing new things, learning, and so forth. But in order to get to those findings, we need research capacity, which in turn needs funding. We need research competence, and many of those who've made it will acknowledge they were in the right place at the right time. A little bit of luck is involved in really finding something noteworthy. And then you publish, and then that generates reputation. And then as you generate more scientific eminence, it's easier to get publications and it's easier to get funding. So we all want a productive career with a virtuous feedback loop of reputation, enabling us to get more funding, more publications, and then do what we really love, which is the science, doing the significant findings. But we can't control the funding decisions. We can't control the reviewers of our publications, but we can't build reputation overnight. And so really our research process, our ability to get more out of the time and resources that we have available to us, is the one thing we do have control over. And so that's kind of the ongoing theme of really what we've been doing at Golden Helix in terms of accelerating the quest for significance. How do we help you find more significant results? So why care about GWAS anyways? Well, GWAS is not dead, despite claims to the contrary. There's still rising revenues in SNP microarray sales. There's nearly 1,000 active NIH GWAS projects, according to this website. There's also new higher density arrays coming out. And custom arrays are likely going to be very important in following up next-gen sequencing studies for cost advantages and others. So even if you're not into GWAS, however, I think the lessons we've learned and we expound upon in the paper apply to the whole field of biostatistics. And what we see is the systemic causes of the mistakes in GWAS still remain. So whether you're moving on to next-gen sequencing studies or you're working in gene expression studies, these issues are alive and well. And so we want to learn some lessons, learn from our mistakes rather than punish ourselves for it. And you know, just as a very interesting publication just came out a couple weeks ago. Some researchers at a pharmaceutical company were trying to replicate preclinical cancer studies. They were taking landmark papers in top journals and found 47 of 53 failed to reproduce. So it's not just GWAS that suffers from problems, and so I'd like to touch on what we've learned in doing GWAS and other studies, and see how we can apply it to doing better in our research process going forward. We don't always think about it, but the prime metric of academia is reputation, and it's a useful metric. It's a useful shortcut for decision making. Students choose what schools to go to based on reputation. The better reputation school you can get into, the better, unless you're into partying and beer, but maybe that's a different kind of reputation. And professors are granted tenure based on their reputation. The system of publish or perish is one we're all familiar with, and that's kind of the world we live in academia. The pro is, of course, saves time and works better than random decisions to decide on funding decisions or publication decisions. In part, is that person know what they're doing? Have they published things that have stood the test of time? But the con is it can penalize those who haven't built up a reputation yet, which could be a newcomer, it could be a late bloomer, or those who may do great science but aren't so good at getting the word out. And of course, reputation is one of those things where it can take years to build but can be destroyed overnight. The funny thing is, though, when reputation is one of the key drivers, you know, in addition to funding and actually getting science done, there's really some devastating consequences in terms of it undermining the scientific method. So, just as a sort of a cartoon of how things are often done, unfortunately, is a search is done for a correlation. Corroboration is looked for. How can I find something that backs up this correlation? And of course, you can look in the literature, especially in cancer, and if you find a given gene, there's almost for certain going to be a publication related to your gene and your phenotype that's going to corroborate what you found. You tell a causal story, publish, and then move on to another correlation. The challenge, though, is in science we really want to understand cause and effect, and if we focus on backing up our story rather than refuting it, we get a lot of, unfortunately, science that doesn't replicate. And of course, correlations are kind of easy to publish, but hard to falsify. If you found a correlation in your current data set, it's reasonable to say, I found a correlation. Someone doesn't replicate it, well, maybe there was something different in their study. But actually positing a cause and effect relationship that's predicted to hold in other scenarios is something that's much more vulnerable to falsification, and hence makes one vulnerable in terms of one's reputation being potentially blemished. So I'd like to talk about a few areas of where we've learned from our mistakes in GWAS, and then talk a bit first about multiple testing. So, of course, publication, reputation, job security depends generally on finding something. Yes, we can publish negative findings, but generally they don't tend to get into the top tier journals. So you'd better find something if you spend $500,000 on an experiment. Well, as a result of that, a lot of early studies in GWAS made type 1 errors. Non-replicable findings were very common. And so the field responded, a new standard emerged, and it was expected that one had to do replication in an independent study. Problem solved, right? However, here's a way that it's reared its ugly head in a similar form. if you run a GWAS, come up empty, oops, I spent $500,000 or whatever it costs to do my study, didn't find anything, I better search harder to go see what I can find. And so then what often is done is, well, we saw some nominally significant findings. They weren't genome-wide significant. Let's take 20 or 40 forward. And then a replication criteria for those 20 to 40 SNPs is used in a follow-on study. And behold, if you do 20 or 40 tests, 1 or 2 by random chance are actually going to replicate at a 0.05 level. And then to further emphasize that replication, let's combine the SNPs from both studies and then show the replicated SNPs reach genome-wide significance, and then tell the story and publish. And I've seen this in papers, and I've seen it in presentations, and I've sometimes raised my hand in a presentation, and they're like, well, yeah, sort of grudgingly acknowledged, but it's really a form of letting type 1 error creep in, and it's no wonder if such studies don't replicate in independent samples. So of course, if we're going to take forward 20 SNPs or 100 SNPs, we need to have a p-value that's 1/20th smaller than 0.05 or 100th smaller than 0.05 to consider replication. I thought it would be fun to rather than say here's what you should do, let's look at what you should not do. And I'm going to pick on gene expression for a moment. We've been doing a fair number of gene expression services projects recently, in having to look at the literature at various examples of studies. And so I'm going to kind of be a little provocative here and say, well, if I wanted to do things as badly as possible, how would I do them? So first, let's make sure we ensure complete confounding in sample collection. So let's pick our cases and controls from separate physical sites. Let's collect them at different times. Let's make sure they're from different populations and different genders and different other demographic criteria. Further, let's ensure once we actually run our experiment on the RNA that we run the cases and controls on separate plates. And then let's add additional cases and controls later, but add more cases than controls. Oh, and midstream, let's also change our microarray platform and do uneven numbers of additional samples on the new platform. Now let's look through 50,000 probes on the RNA array, or it could be an RNA-seq experiment, and let's find the 50 or so that are highly significantly differentially expressed between cases and controls. And then let's do a cluster plot to show, voila, the in-sample clustering beautifully separates cases from controls after we've selected through 50,000 markers to find 50 that highly differentially are expressed, and could be due to chance alone. And then let's not search for sources of experimental variability that could explain away our findings. And then finally, let's not make our data or analysis transparently available. So you might laugh at this, but there are examples of all of the above being done. And certainly some of the above regularly happens, probably I'd say 80-90% of the time. It's hard to find experiments where this doesn't happen. And there's sometimes reasonable reasons why we can't make things perfect, but we need to really be aware of what that's going to do to our analysis. Let's look forward a little bit in next generation sequencing and ask ourselves, are we going to have a bit of a wake-up call with type 1 error? So as you know, there's many exciting published success stories of next generation sequencing finding the causative variants for rare diseases, especially with recessive inheritance, perfect penetrance, and often pedigrees. Usually nuclear families or even trios are used. A whole series of filters are run, and here's kind of a list of them from our SNP and Variation Suite software manual. What goes into that whole filtering process is kind of a classification, a prioritization of different types of mutations that could be at various degrees of potential rarity and being non-synonymous, deleterious, etc. We make all sorts of assumptions in terms of filtering, like often we're going to just look at the exome, for instance. And the amazing thing is, looking at a very small family, one can actually narrow down to a handful of variants and look at the genes, see if they make sense, and potentially describe the cause of a condition. But I had a very interesting discussion with somebody at the American Society for Human Genetics last fall. And this fellow had a poster. I don't even remember his name, but he had a very large pedigree in some disease. They were looking at how it segregated. They had 13 affected members of the family, so this was a big pedigree, and then plenty of unaffected members. And they found a variant after their filtration process that was in 12 of the 13 affected members and none of the healthy members. And they wrote their poster up and said, aha, we think we've got it. And there's a nice story to tell about the gene that this variant falls in. And then he said, well, since we published this poster, we found another variant that was in 13 out of the 13 affected members and none of the healthy individuals in the family. We now think that that's the answer. So if we can get that wrong when we have 13 affected members in a pedigree, and imagine if we had just picked 4 out of those 13 or some subset nuclear family, how many of our nuclear family studies are really problematic? And so really one of the things we're going to will probably become one of the standards is some sort of functional verification is going to be imperative. One of the challenges, however, is mice models are not going to be realizable for many of the genes that we find. So we'll see we'll see how this all fleshes out. So I'm going to talk a little later in the presentation more about the scientific method and some other topics from the paper, but I'd like to do a quick poll and just see if everybody's awake, and ask you, have you read the biostatistics article? And I've just clicked, and you should now see a poll on your screen, and I'm wondering if you've read the article. No, and you'd like us to arrange for you to be able to get a copy, or no, who has time to read papers? So I'm just watching the votes come in, and I won't skew the votes, but just leave another 30 seconds or so. We've got 70% of you have voted, and 77%, 78%, 20% are doing real work. Well, actually, all right, well, looks like everybody's almost voted. I'm going to close the poll in a sec. We've got about 75% would like a copy. 21% have read the paper, so I'd love to hear your feedback. And 4% don't have time to read the paper. So thank you so much, and let me continue with the presentation. Click. All right, so let's talk more about the impact of poor study design, and I'm going to skim lightly over some of this content because I have talked about it at length. Many of you have not seen my former webinars on this, but there's a nice one that you can link out to here that talks all about study design issues, and there's a couple others on quality control and so forth. But really, this invited editorial that we wrote for biostatistics originated in a blog article that was talking about how 93% of the studies we'd looked at, that we personally done analysis work on, had experimental design problems, and we kind of raised the alarm, and we were invited to do an editorial to expand on this topic. And so there's so many ways in which things can go wrong. You can have unbalanced plating of cases and controls, borrowing controls from another site, which we'll talk about later, trios split over plates, multi-site genotyping, not randomized by phenotype, and the impacts are huge. I can't tell you how much time we spend fighting with data that has not been where the study was really not designed with the hypothesis in mind, and for one reason or the other, the proper randomization didn't occur, leads to huge amounts of analysis time, throwing away of data. Type 1 error is a huge problem, endless struggles with batch effects, and one of the key things, it's something, this concept that if you think of a research process, the errors that occur early in the process, and experimental design is the very beginning, they amplify and are more costly to correct over time. This is something Deming, who's the father of the quality movement, observed. And so it's like the leverage point to getting better results in your scientific research is right at that place of study design. And so I've talked in the past about the huge batch effects in the Wellcome Trust study, where cases and controls are both collected from different sites and then plated separately. Two complete confounding steps. We were just learning early in the field, we, the royal we, but and so it's understandable. But what happened is, as we noted in our article, a whole set of quality control filters came into existence as a way to basically remove the effects of confounding, or at least try to ameliorate them. And the truth is, they're a band-aid that is just not 100% effective. You see QQ plots are ridiculously huge type 1 inflation in the Wellcome Trust study. By the way, the 2 of the 30 studies that actually were designed and carefully randomized, one was a Alzheimer's study that GSK had collected from about 9 different sites, and then carefully randomized the sites and the case and controls over the plates. And without filtering a single SNP, they have a beautiful QQ plot, and their APOE SNPs are highly significant as expected. You shouldn't have to do filtration if you've done a good job at both sample collection and randomization. Those are in our arsenal, as a fallback. But unfortunately, I think the Wellcome Trust sort of set a template, and then ever after, everyone sort of runs a set of filtration steps as if that's the way a statistician should handle outliers. And so here's a Manhattan plot where you have huge numbers of false positives in the Wellcome Trust study versus this GSK study has none. And then you can look at the C and Vs, which the intensity data is even more skewed, where you get massive inflation when there's complete confounding, and yet it's pretty much under control with a well randomized study. And you can see here the plates is one of the real big effects in genotype error. This is the Alzheimer's study. Each plate kind of clusters differently when you do a principal components decomposition of the variation. But when you look at color coding case versus control, you see it's properly randomized over the entire data set. Perhaps there's maybe one plate here that's a little over skewed towards either cases or controls. But then what happens is when you run the association test, those random errors due to plating, etc., are sort of come out in the wash. So I'd like to segue. I'm going to talk shortly about the Sebastiani retraction, but I thought it would be very good to first talk about, that we do not want to be in a mode of criticizing the people when mistakes are being made, but rather learning from those mistakes, and let he who's without sin cast the first stone. And so my first paper was in Nature as an undergraduate, and I did the key research that was in error. Some astute readers noticed that I had misassigned some of the NMR spectra in terms of which signal came from which peptide. And they graciously notified the main author, and they worked to address it. 4 years later, an erratum came out, and fortunately the structures still did hold up mostly, and retraction wasn't called for. But I still wince about it. My first paper, Nature, in fact my only paper in Nature, and it was marred by these types of errors. So then, the question is how to learn from our mistakes instead of punish ourselves and especially others for it. So I thought I'd set that as a let's not think any of us can be immune to making mistakes. In fact, really making mistakes is how we learn. So the Sebastiani retraction, their paper is, of course, Genetic Signature of Exceptional Longevity in Humans. Here's sort of the sequence of their papers and expressions of concern, retraction, and so forth. And what happened was they had used different genotyping platforms and neglected quality control steps was sort of the criticism it received. And a lot of attention was focused on, that there was this array platform. In this case it was an Illumina chip, or a couple chips. That one chip had artifactual genotypes for some subset of the SNPs used in their predictive model for longevity. But there wasn't really a focus either on the criticism or even on the subsequent paper on the experimental design flaws that are inherent. The truth is there's error in every array, and artifacts happen, and the real problem was how they divided their cases and controls among platforms. So anyhow, they looked at their SNPs that were used in the model. They went to apply quality control steps to remove the worst offenders. They validated SNPs via TACMAN and redid the analysis. Now they stood behind the revised results. They found they still could get good predictions, but science still had enough questions and that they required a retraction, but no misconduct was considered. And then they published the revised manuscript this year in PLOS ONE. So really, just looking from a scientific method perspective, the authors should be commended for making a bold, falsifiable hypothesis. Look, we think we can predict exceptional longevity using a set of SNPs. That's pretty exciting if it was true, and they're saying, look, and we could extrapolate to potentially other populations, and this could be a useful tool. But of course, the bolder the hypothesis, the greater the scrutiny, and so I think there's some serious flaws that just couldn't be addressed without doing an additional experiment, and of course that's costly. So the key thing is their cases were drawn from 3 independent studies, but the controls were all drawn from this Illumina iControls database, and that's a set of SNPs from studies that various people have just deposited with Illumina. It's not the DNA, it's just the data. You don't know exactly how the data was processed, which versions of software and so forth, or what were the experimental differences that could have been peculiar to whatever subsets of the iControls were selected. And so the claim that it's replicated in 2 independent studies is somewhat dubious, because iControls is really, if you will, from the point of view of confounding due to arrays, the iControls database is, if you will, one population. And so the controls are completely confounded in 2 ways. Some of them came from a different Illumina array, and then all of them came from separate sources than the cases, with all sorts of unknown sources of variability. So then, the devil's really in the details of trying to rule out that confounding, and what they did with their TACMAN assay was they looked at 30 of the 281 SNPs from the cases. Now they don't have the iControls, but they did look at different arrays, and they found that they had 99% concordance when they looked at least at 28 out of the 30 SNPs actually perform well. Now one of the 2 SNPs actually had 93% call rate, and the other failed the TACMAN assay. So if 1 out of the 30 had problems with TACMAN. It's hard to say what the problems also could be with the genotyping array for the other ones. Now, the authors have concluded they've ruled out array bias, but is this decisive? I mean, they've done some good homework on this, but there's still room for doubt, and this is the problem with experimental design. So the fact the samples were tested from the same array as the controls, it's not the same as testing the controls themselves. And here's the kicker: an average high accuracy, that you've got 99% concordance and really good call rates, doesn't rule out bias in a multi-SNP prediction panel. You can combine many things with 99% error and maybe a few that have 93% error, and that random error, when combined all in, non-random error that's potentially correlated with the site or the platform, in this case that they all came from the iControls database, the controls, that could combine in a complex model to be something that's highly impacting the prediction. And so, I mean, we've even done a study with someone who had run purely TACMAN assays on cases and controls, but they did their cases and controls at different times, and the pattern of missing values of the TACMAN assay was beautifully correlated with whether you ran the cases or the controls batch. And so he had something like a 95% accuracy in differentiating cases and controls purely based on missing values with a TACMAN assay. So, we're still left with uncertainty, and it's probably why Science had enough questions to not publish it. And really, is it an iControl classifier or a longevity classifier, or maybe some combination of both? Really, a truly independent replication experiment is required, but of course that costs money. And I just should make a comment that there was a lot in the classifier development process in terms of doing proper training and testing, not looking at all the samples when you do feature selection, but holding samples out. This really, that methodology of doing that is very well described in the MAQC Consortium article. I was one of the many co-authors on this Nature Biotechnology article. So if you're thinking of doing prediction, it'd be worthwhile looking at some of the guidelines from this paper. Now, in the author's favor, there is an anomaly that suggests this is not all platform or error or confounding, in that the prediction works better for extremes of phenotype, and so you wouldn't expect control confounding to explain that, but it is possible confounding in case collection or experimentation could. And so one would need to really drill carefully into is there some way that the really old people were handled in terms of their samples, or that they were potentially put on a different set of plates, and so you'd want to rule that type of stuff out. So really, the moral of the story, let's use it as a learning experience, and say we've all been there, or wish or hope we won't. Is that flawed experimental design, especially complete confounding, which is what was going on here, will throw really a sticky, undispellable shadow of doubt on your results, and that really, frankly, is a bummer. So I'm going to skip over this really lightly just for the sake of time, but really, I think it's come out in me talking about some of the problems with the Sebastiani experiment, but there's so many sources of data collection, plating, and genotyping variation that if we don't control for these, both in the design of the experiment and the execution of the experiment, the design of the sample collection and so forth, we'll get into real trouble. And if you'd like to hear all about this, my earlier webcast, you can link out to it and hear some more. And in particular, one of the skills that's particularly important is understanding how to block randomize on plates, and that webinar talks about how we've done it. It's one of the services we do as a company, and have done it for a number of experiments, including including Wadsworth did a large Parkinson study that's now deposited dB gap and was published in Nature Genetics recently, where we did the randomization by site and DNA extraction method and case control status and I think another parameter or two, and made sure that there was a beautiful randomization of all those potential sources of confounding across plates, so that the uncontrolled variability due to multiple site collections, so forth, you could potentially factor it out. And it turned out that you needed to do no SNP filtration on that study. Any highly significant findings were you would not want to look into them one by one, and you didn't have a bunch of inflation type 1 error where you would actually have to resort to automated filters. So I'd like to do a second poll, which is to ask you all, given Golden Helix has got so much experience with this, and so much pain and suffering can happen in terms of really, especially setting up an experiment and thinking about subsequent analysis, I'd like to ask if you are interested in a free consultation with our services team, where we could potentially advise you if you're thinking about doing a project or getting rolling on one, or have made some of these mistakes and are curious what can be done about them. Whether you'd like to explore a services project proposal, I'd check this button if you really got something urgent. We do have really limited bandwidth right now, but we'd be happy to talk with you about if you've got something pressing, that grant needs to get out the door, or that paper needs to get out the door. And finally, we recognize many of you have no interest in working with us, but and that's fine too. I should say one of the interesting things about how we've worked in our services is if you ever worked in a situation where you send your data off to a black box and get back results. We don't work that way. What we found is the best way to work is to bring the expertise of the investigators together with the expertise of our analysts, do regular webinar-based meetings, so that as you do projects with us, because it's not us just doing the project, we're learning and doing the project together. You learn. We both help you catch the fish as well as how to do the fishing yourself next time, and so that's one of the real benefits of working with us with all the experience we have. All right. Well, thanks very much, everyone, for the poll. I think I'll close it. Well, looks like 63% have voted, so maybe a few more might have been pulled away, but I'll close the poll here. So back to the webinar. So the next thing I'd like to talk about is the scientific method, and really, we're all scientists. We think we know what science is all about. I realized, I've never been taught a scientific method in school, and there's so much that we've seen that actually points to really not knowing about the scientific method as one of the sources of poor experimental design. Now, I thought it was very interesting. If you look at the top Google image for scientific method, and this comes from a resource site that's pointed to by national science fairs as a source of information on how to do your science experiments. That's why it's #1 image. You have this idea of constructing your hypothesis and then seeing if it's true, and true or false. Whereas we the point we've been making is really science is all about falsification. And so I'd like to try to demonstrate in a kind of a tactile way, and maybe this is just ho-hum for some of you, but by all means, have your students look at this if they've never really been exposed to really what the scientific method's all about. So I'm going to look at generating a hypothesis about the behavior of a time series. So I've got some variable y that changes, goes up over time, and I've got 10 time steps. And I have a hypothesis that there's a linear relationship between y and time. And lo and behold, I fit a line, r squared 0.996, a p-value of 7 times 10 to the minus 11th. Man, this really looks linear. I'm confident it's time to publish. Well, with the scientific method, we have to say, well, what's the predicted effect of this relationship if we were to look forward in time? Well, let's look at the next 20 time steps. Oops, our hypothesis is falsified. It's not linear at all. In fact, it looks much more consistent with a quadratic relationship. Y is t over 8 minus t squared over 250. R squared 0.997, p 10 to the minus 38th. Wow, that's significant. It must be true, right? Well, let's look at some further time. Oops, it's not quadratic. It looks like it's a sine wave. Oh, and it's a real simple parsimonious explanation. It's t sine of t over 10. That's so much simpler than this quadratic equation. Must be true. Look at that ridiculously significant p-value. Well, it turns out the truth, because I created the time series. Trust me, I have a PhD, right? And I had my graduate students check it. It's a damped sinusoidal with a very light damping that you wouldn't see without looking over some long time period. And so you see the divergence of those hypotheses. Here's the linear hypothesis, the quadratic hypothesis, the sinusoidal hypothesis versus the data itself, which is a damped sinusoidal. And so this is kind of trying to demonstrate this idea. We can never say, I know for sure this is true, I've proven it's true. And so what's really funny, though, is how the concept scientifically proven is used throughout advertising and public discourse. We copied a couple funny ads. Here's increase your IQ by up to 20 points in 20 days. Scientifically proven, money back guaranteed. Wow, I want that. Science says it's so, right? And instant inch loss is a weight loss program that's been scientifically proven. So until I'd written this paper in biostatistics, I hadn't really been exposed to philosophy of science, and you really got to kind of spend some time with Karl Popper and in his book called Conjecture and Refutation and other works, where to understand really what is the foundations of science and the scientific method. And of course, there's people who say there's the scientific method as defined by Popper is not strictly what scientists do, and that's fair enough. But there's an important point about falsification that really needs to be made. So the metaphysical foundation of science is there really is an objective reality. I can knock on the table. There is the way reality is, and really truth is the way reality is. It's the nature of reality. Actually, turns out science in any metaphysical thing, whether it's religion and so forth, is actually, there's a bedrock base that is actually not falsifiable. So it's not falsifiable that there is an objective reality. Life could all be a dream in which it seems that reality is the way it is, and we could wake up one day. But in any case, it seems it tends to be pretty a pretty solid metaphysical foundation for those of us who are scientists. But there are other metaphysical foundations one could choose, and Popper actually talks about how can one choose between metaphysical foundations. Is life a dream? Is what is a better foundation? So really the scientific method is all about investigating the nature of reality, and we do it through observation. We hypothesize from what we can see, so subsets of reality, and then we generalize outside of what we've observed to more and more universal supersets. And then you get things like E equals mc squared, this actually universal statement about how energy and mass are related for all all space and time, if you will. And so those are, if you can say something about the entire universe, then you need to just, and in order to say it's true, you'd have to look at the entire universe to say it's true. And so you just can't do that without a method to verify the unobserved, which apparently, science really doesn't have a method to confirm what we haven't observed. So if you think about reproducible research, the idea is I need to be able to set up a scientific experiment that I can do, that somebody else can repeat, and it generalizes out of the particular laboratory setting that I have, and can be recreated in another person's laboratory setting. And so while we cannot confirm something is true, especially a universal statement, a single counterexample can falsify it. If we say all ravens are black, if we find one albino raven, we've falsified that conjecture. And so science then is really a process of conjecture and refutation, not conjecture and somehow proving. But unfortunately, it's something that's kind of deeply ingrained in the public mindset, and in fact in some of us, even as scientists, at least. Even though as we may espouse that, often our language actually talks about certainty and knowing and truth and so forth. So the method of science is about really we conjecture something that covers some swath of reality, and then we eliminate that, and by essentially this process of elimination, we approach truth and yet apparently never reach it. And so the scientific method, we observe reality, form ideally a hypothesis that's causal to explain our observations. And then the key thing is think of predicted effects, as I showed in those line graphs, that are not already been observed that would be consistent with your hypothesis. So make sure you're not just sort of trying to explain other things, but you actually are trying to predict something that you haven't thought about until you came up with your hypothesis. And then the more unexpected, the better. Design an experiment that falsifies a hypothesis, i.e. that shows there's a contradiction between what you expect as your predicted effects and reality. Now, once you analyze the experiment, if you see a contradiction, you reject your hypothesis and go back to forming a new one, but make sure the new observations are not thought of as predicted effects, that you're now basically creating sort of a bunch of exceptions to your theory that kind of make it all work, as opposed to really coming up with new predicted effects. Now, if contradictions not observed, don't quit there. Try to repeat several times until you've exhausted the limits of your ability to falsify, and finally publish and let others try to falsify. And of course, that's just what happened in what happens all the time when papers are challenged. And there's something I really wanted to draw to everyone's attention. There's this classic article from 1890 that was republished in Science in 1965 by a guy named Chamberlain, and he said, the problem with humans, with scientists, and all of us, is we get a parental attachment to our hypotheses. You form something and you're like, oh gee, it really explains things, it really seems good, and you become attached to it, and it becomes harder to kind of think of ways to falsify it, because I want to publish this thing, and it seems really exciting and interesting, and so you can take and tweak this scientific method to use his method of multiple working hypothesis, where he says, first form a bunch of competing hypotheses to explain your observations. And you're going to not have as much parental affection, because a lot of these hypotheses are, especially if they're mutually exclusive, some of them are going to have to get knocked down. And then you design your experiments to falsify as many hypotheses per experiment as possible. And apparently, there's an article that refers to this article. I'm forgetting the name of the author, again in Science, who talks about Crick's blackboard, that they would build a whole decision tree of potential hypotheses and think about ways to knock down as many. This is kind of the something that thinks has been lost to us. And so then you essentially follow a similar process, but it gets you out of the sort of the blind alley of parental affection for your hypotheses. The thing is, the scientific method, there's some reasons it doesn't, there's some challenges in biology to its application. So the idea of a controlled experiment, there's a real problem with that in terms of human beings. We can't clone human beings like we can clone fruit flies. And so there's so many sources of variability uncontrolled. And often we end up having to collect hypotheses generated post-data collection. So most studies are observational with humans, where we don't have the ability to control things. And this Latin concept of all things being equal, ceteris paribus, this idea that we look at human beings as identical objects, and then we're going to say, okay, salt is bad for everybody, or smoking's bad for everybody. There's this idea that we can make a universal statement with entities that are not equivalent is a challenge in applying the scientific method. So we start statistically trying to make statements about whether something's more likely. There's a challenge of direct observation, especially over time, and looking at the time behavior of microscopic systems. And I think, as I've been saying in a number of my blogs lately and in this article, longitudinal probe test of individual people may well be the paradigm that is going to replace looking at large populations, that each person is a unique system, and we're going to be looking at the behavior over time of that unique system to understand cause and effect. So finally, I'd like to close with just a few more thoughts on how we can do better. Really as a sort of a summary, we really need to think about designing our experiments to falsify our hypothesis. Think about, during data collection, your hypothesis. Obviously a lot of interesting data collections gone on, like the Framingham study and others, where we're going to generate new hypotheses later. But think about the categories of hypotheses that are going to be done in the future, and how one can really design data collection and then subsequent experimentation to minimize those sources of confounding that ultimately are such a headache. So we don't want to stop at correlation finding as a theme that's also amplified in the paper. If you think of science, it kind of moves from classification. We create names and categories for things. We move on to comparing them, and that's correlation. And then we move on to cause and effect. What's the sequence of cause and effect that describes a system? You could look at the method of multiple hypothesis as a way of looking at multiple chains of cause and effect and deciding between competing ones. And then science moves up into aggregating those into theories and then comparing theories, and then sequences of theories move into entire systems of knowledge and fields. So correlations are easy, but there's only so much understanding we can get about the nature of reality. If a finding is too good to be true, it probably isn't. And we found that time and again, your first cut analysis of a study, especially when there's confounding, wow, there's some beautiful signal there, and you get all excited about it. But then if you're diligent and really look to falsify, all too often they get knocked down. And so when we're attached to the outcomes of experiments, I think it gets us into trouble. And really, I think reputation will follow from research process excellence. It is the thing we have control over. If we can have the highest degree of integrity with how we do our experimentation, our analysis, and making available our data and our analysis process, so that we can really address some of these real challenges with reproducibility in the biological sciences. I think we're going to help ourselves in terms of our careers, but also help the larger sort of field of human endeavor of attempting to make all of our lives better through improving human health. And finally, I think the real message I'd like to leave us with is we really learn through our mistakes and the mistakes of others, and it really, we need to think about it as we walk around forming mental models of reality. And it's when our mental models, our expectations of what isn't what's going to happen next doesn't happen, that we actually revise our mental models. And so when we punish ourselves for our mistakes, I think we get into a blind alley of stopping learning, and then that's going to impede really the application of the scientific method and all of the benefits that come out of it. And so I've expanded on this a little more in my blog, Learning from Our GWAS Mistakes, From Experimental Design to Scientific Method, the same title as the paper, and you can click out to that. And please, by all means, comment. And if and the 20% of you who've read the paper and those who are going to read it, I'd love if you would comment and we could have some dialogue on the blog, and I'll be checking it regularly and look forward to your thoughts. And by all means, come up with ways in which you think that my hypotheses don't fit reality, because we're on ever quest to learn more and go on that journey of discovery together. So now I'd like to swing over to Q&A. We've got, as I said before, you can click and type questions in the question panel, and I will try to get to as many of them as I can. And we're getting close to the top of the hour, but I'll be happy to hang around as long as you'd like. And we will have this recording made available later, and would love to start answering your questions. So, someone asked, I think the main problem is the system. It fosters this culture whereby many people do not really care about the methods or results, only getting data for publications and funding. Yes, absolutely. And really, I think, the message I've been trying to deliver is it's not the people, it's the system. So let's fix the system rather than fixing the blame. Reputation as the prime metric of the system, when really our goal is about scientific output and advancing human knowledge, or even if we're funded by federal agencies that are in turn the taxpayer expects benefit from, we should be looking at how do we create measurements that deliver bottom line value of life and health to society. So someone asked a naive question, how batch effect occurs. From this talk, it's due to non-randomization in case of controls on plates, but why? So I talk about that in some of the other presentations. But basically what happens is, if you think about the fact that things can change over time, and if you run one set of samples at one time period and another set at another, there are going to be experimental differences. The temperature of the ambient atmosphere could affect binding of molecules. I mean, these arrays actually involve chemical processes that could be affected by thermodynamics. We found there was a paper by Signature Genomics finding that ozone concentrations in the atmosphere affected the performance of their microarrays, and how they noticed it was that the error in their arrays went up and down with the train schedule. Their lab at one time was close to a train station, and so they put ozone hoods over their instruments to remove that source of variability. So there's just so many sources of variability. I think why plate-to-plate variability is so big, I suspect it's in the amplification process, how much time that goes. There could be different quantities of DNA. RNA experiments have other considerations, and we can never assume that we're going to get perfect measurement, and so that's why it's so important to randomize. Okay, so let's see. Scientists are often not aware of pitfalls in the genotyping process, such as plate effects, etc. Are there standards for labs conducting such processes so that researchers are not victims? I do not know of any, but you know, I'm a little removed from the actual lab work, and so I apologize. I don't know the answer to that question. And so, how can DNA extraction method be confounding for genotyping? So the DNA extraction, so there's what is the source of DNA? So I've seen studies where the cases were from blood, and the controls were buccal or saliva, and there's just all sorts of performance characteristics of the arrays on those different sources. In fact, there are some differences in the cell types and so forth. Also, I mean, who knows? Maybe one site sort of left the samples out on the counter for a few hours longer than another site, and certain reactions took place due to it sitting on a warm counter. So there's stuff like that where and then there's the actual whatever stabilizers and so forth might be put in with the DNA. If you're using a different kit, there just could be slight differences. And remember, we're dealing with atoms and molecules here that can be perturbed by the slightest thing. Fortunately, DNA is quite a stable molecule, and so we're not without some hope of having consistency. So someone asks, we analyze our CNV data, we correct for batch effect, DNA collection site, DNA source, normalize our intensity signal using our own data, not HapMap data. However, we still need to use 90 principal components to have a nice QQ plot. Can you help us how to correct the confounding factors using our software SVS? Well, what we found with studies is there's just only so much you can do to correct the data, especially CNV studies. And we could talk with you one-on-one about this, but there is not a magic bullet to correcting confounding, especially complete confounding. In fact, it's in many cases the experiments become a wash. So this is why we keep emphasizing the design up front. And we indeed have developed tools to try to mitigate it, because unfortunately most customers do have these problems. And what we found is sometimes you just have to live with that level of noise and look for the very large CNV variants that will be less impacted by batch effects. And that's, I think, why we see so few papers of CNV GWAS as opposed to just finding a few large variants. So, someone asks how I think we can deal with publication bias. For example, the American Journal of Human Genetics has a policy of not publishing negative results, and they told this caller that. Well, I don't have it at my fingertips, but there have been some good authors who've talked about methods for this, standards for how we could address this, and I unfortunately just don't have them at my fingertips. Some people have said, the whole publishing process is just waiting for somebody to figure out a killer app to solve these issues, and I suspect that's probably what's going to happen. I wish I had a better answer, and we do talk a little bit in our article about some guidelines on reporting observational studies, that there's a standards body that's come together and come up with some guidelines. And so, but in terms of not publishing negative results, that's a real problem. And I'm glad you raised that issue. So there's no publication and rewarding mechanism for successfully falsifying other published hypotheses. What can we do? Yeah, and in fact, it's often thought of as you're threatening, the other party that wrote the article by actually pointing out the flaws in it, which unfortunately is an outgrowth of the system. And if you think about it, if science is about conjecture and refutation, if you kill our ability to refute others, you basically don't have the advance of science. And so, sometimes people just need. It does turn out, though, that the more famous there's a publication. Gosh, I wish I had the name at my fingertips. They talk about how initially, I think it was Ian Eadie's article actually, and it's cited in my biostatistics article. He talks about how initially you get an interesting finding, and then a bunch of people look for corroboration, and then they publish, say, we saw it too, but the people who saw falsifying evidence typically don't get published. And so then the finding sort of gets famous, and then there's actually reputation benefit for somebody to start publishing falsifying evidence. And so you actually can, develop reputation by sort of being a giant slayer, but only really after something's kind of gotten enough of a snowball running of being sort of the standard knowledge in the literature, and then what you find is the subsequent publications start falsifying it, and then we start saying, well, gosh, that finding seemed to have evaporated. So someone asked, is there a chip that you recommend? And I'd say no, we don't make any particular recommendation, but there's a lot of good platforms for different uses, and we've in some previous talks talked about considerations in choosing a platform, but I probably can't really go into all that right now. The question is, GWAS seems contrary to the scientific method. Rather than attempting to prove a hypothesis false, GWAS performs many tests and looks for positives. Your thoughts? And this is an interesting thing. We talked about it in our article. It's actually, you really are starting with sort of a broad general hypothesis that if there is some sort of genetic factor that differentiates between cases and controls, and you're actually falsifying all of the insignificant results, and then the ones that are left, they're not verified, they're just not falsified, and then they become candidates for further exploration, functional analysis, etc. So it's not contrary to the scientific method, but it is sort of it's sort of a signal hunting thing, and we talk about in our article about how Fisher himself, R.A. Fisher, was critical of hypothesis testing in that he said it's really, I'd have to get the exact quote, but he said something to the effect, it's really when you don't know anything about something and it's sort of a blunt instrument and you use it, and so that's kind of where we were in we didn't know where to look, and so it actually is a very useful instrument, but then we need to go deeper into causal explanations as for our follow-ups. So someone was not aware of this, and I'm not sure what they're referring to, but like MAQC, are there any seminal papers that show through deep analysis what should be the proper standards for GWAS studies? Wow. I know there was a good paper by Bruce Weir that talked about methodologies for quality control, but it wasn't about sort of the design and setup of the experiment. And I think if there is one, I'd love to hear about it. If someone could email me, I would be happy. Especially if you authored it, I'd be happy to communicate it to others. And potentially, if you'd like to give a webinar and talk about what you think are, if you've got a published set of standards, we'd love to potentially provide a forum for you to talk about it. Someone says, and we're getting, that there's a growing fantastic body of publicly available data genotyping, expression, etc., that they want to be able to use. What's your advice for handling that data, or do you avoid it? So yeah, so this is this thing about, can I borrow from other studies, and the advice I'd make on it is, the best when you're borrowing from another study is if your initial study, and say we're talking a case control study, but it could be a quantitative phenotype or something else. Suppose you have a balanced 50-50 mix of cases and controls. If you could find a second study that also had a balanced mix, then you're on a much firmer ground for combining them in a mega-analysis. But if you have a second study that's only cases or only controls, or the proportion is very different, then you get into a lot more of those questions of confounding, like we talked about with the Sebastiani paper. Now, even if you are going to completely, borrow cases or borrow controls, there's nothing to say you can't do that and then go back and validate all of the SNPs using another method once you think you've got a particular finding. And so it's not that you can't do it, it's just you've got to really be aware of the risks. And of course there's real cost advantages. In fact, this was one of our reviewers of our paper was making the point that value has come out of these things, and there have been ways to sort of to address the issues. And that's absolutely the case. Good findings have come out of studies that had all sorts of problems with experimental design. It's not a total loss, but the point is, it doesn't cost much more to think about how to do it right, and then you'll find that your study's so much more defensible, and you don't have to resort to all sorts of quality control acrobatics. So we've got a few more questions we'll answer, and we'll wrap. So what design flaws have you seen in the Wellcome Trust case control consortium study other than genotyping errors? And so so the design flaw was that if you're going to compare healthy controls versus affected individuals for a disease, you want to control the sources of variability. So they had collected their controls from one site, and then there was a second site, so there are actually two sets of controls, and those were from different labs in all the cases, and that's maybe necessary, because obviously there's going to be disease specialization and so forth. And so that was perhaps unavoidable, but that, in terms of if you're designing a prospective trial, for instance, what you would want to do is be collecting DNA or RNA, whatever that trial is, from ideally equal proportions of cases and controls from each site in a multi-site study. Then when they did their plating, they put sometimes they mixed the cases for different diseases, because remember there were 7 different diseases. They would mix them on plates, but they didn't put the controls using a block randomization approach. Now there was some rationale for why they did that, and there was some interesting discussion on one of my blogs with one of the people who worked on the study, and they were wanting to get the controls done earlier, I think. And so that was one of their considerations. And of course, there's also some other issues where if you look at the age of the controls, a lot of them were kind of young, and so when you're looking at diseases like type 2 diabetes that could happen later in life, there's some issues there. So just those are some of the thoughts on that. So here's a comment. Researchers generally submit samples for genotyping already placed on plates. On the other hand, the genotyping lab doesn't have access to the phenotypic information in order to replate the samples the ideal way. So it's really up to the researchers, though the genotyping lab could remind the researchers. So absolutely, this is what usually happens, and this is why we have this problem. When we did the design for the Wadsworth Parkinson's study with Heidi Payami, we worked with the lab to actually ensure that plating occurred. And actually, they insist on, I think it's Coriell actually. No, sorry. Coriell's where some of the controls came from. Oh darn, I'm running into brain fog. I need some more coffee. But the lab that did it highly encourages having that randomization done. And you'll find, there's some reasons of patient confidentiality where you don't want to give that information to them. There's ways that you can essentially still blind it, but give them a manifest that says this sample, this barcoded sample goes here, that barcoded sample goes there. You don't ask your core lab to do the experimental design. You got to control that, and then you basically give essentially the coordinates of where you want what put, and there's robots that can do that type of stuff, or pipetters. So someone thanked me for the talk on how to regarding sciencing, and that we all need the reminder. Thank you. I found until I wrote this paper, I really was myself fairly ignorant of the scientific method. And so someone says, thank you very much, Dr. Lambert, for a very useful webinar. One question, I'm using genome-wide SNP data from affected and non-affected pedigree members from ethnically diverse genetic isolates with aggregation of same complex disease. MDD, SCZ, etc. I try to evaluate rate of genetic heterogeneity of the disease, related gene pools diversity. Is there any such option on SVS that I could use? Yeah, there's one approach people use is the Eigenstrat methodology, where you do principal components analysis after selecting appropriate SNPs, and that's something that we could assist you with if you'd like to contact us. Yes. So the lab that we worked with on Heidi Payami's study was CIDER. So they they were very helpful in working with getting that plating right, on getting appropriate randomized cases and controls, as well as putting appropriate replicates. So someone asked, family design, should we put all family members in one plate? Well, it gets to what your experiment is about, but generally the answer would be yes, if possible. And if not possible because the family is so large, then you'd look at how you can ensure that affected and unaffected individuals are evenly divided if your family actually has to go over multiple plates. Well, thank you very much, everybody. That's the end of the questions that have come in. And again, I'd love to encourage you to read the paper, comment on our blog, and we'll make this presentation available. Let your friends know about it if you feel it would be useful for them, and we'd be happy to help you in any way with really trying to advance your research. So onwards and upwards. Take care.